So Isabella, I believe, has already shown you how we do crossing, which is the beginning part of the breeding program. So Isabella, Dr. Mohammed Iqbal is the research associate who does much of our crossing, and Isabella helps as well. And she is working in the quality lab and the disease-resistant nurseries. So what we are now, because there's been such a lot of rainfall this year, it's not really easy to go where we have our F1 or our populations that are the beginning part of the program, but they're all growing in this area around here where we have irrigation. And we're near the building of F62 at the central part of our station. So the F1 crosses in a breeding program go on. We then grow the seed from that cross, 50% of the genes from the mother, 50% from the father. They're grown in a field of small rows. The rows could look something like this. You see each one, what you're looking at in this particular field is about 50,000 little plots, hill plots, of separate genotypes. And our F1s are growing in a similar outlay. We plant them by hand and they're growing way beyond a big puddle over there. We also harvest that F1 seed and we grow it as the F2 generation where it has the most genetic variability. And we select a little bit in that generation for maybe things like height and disease resistant. Then we take seed from that, send it to New Zealand, do the same process. We grow them in population structures that might be 25 meters, 2 rows. In the F2, we might grow 8 or 10 rows of that population, and we narrow that down as we start selecting heads out of that. Till we get to this stage here. And in this field, what you see is about the F5, F6 generation of 40,000 odd plots. This field has been infested with 3 types of rust. So it has had 3 types of rust sprayed on spreader rows. And I thought I was standing next to a spreader row, but here's one here. And this particular row has very susceptible genotypes. And we spread our disease by spraying that row, which then serves to spread the disease around the field. And in a few weeks, the disease is already in here, but we've had so much rain of late, it's all washed off. And in the spreader row, you see leaves that are turning yellow and stuff, but the actual rust disease is washed off. And of course, there's all sorts of other diseases that build up in this field because we irrigate it, create a mist, create humidity, which leads to fusarium if there is any prevalent, etc., etc. But what we do in the breeding program now is out of 40,000 rows on this side here, we will select about 4,000 or 5,000 of them to go to New Zealand, 1 head out of each of these little hill plots. If they're short enough, and that particular line is, you can see that in relation to a line that's too tall. And this little area here of the 4 rows here would all be progeny of 1 cross, and you can see the variation in height. And you can see that this is an awnless times an awned cultivar with very different heights, and that creates babies that are very different as they approach homozygosity. And so while these are not purely homozygous lines, you'll see that they show roughly what they're going to be like when they become more and more homozygous. So there's a nice short line that looks really nice. It has a gene called LR34, which confers resistance to rust in it. This looks like, and then we will select based on its reaction to rust and how early it is. And those lines are all babies of 1 cross up until you see the 2 barleys there that we separate the field in. And that's another cross behind it, which looks to be a more uniform cross between 2 short awned types. And you can see it in behind there. But in this field, we then select, and this is the last field it grows in Edmonton in Alberta before it goes into the yield process, or the yield process. So we got about the F7, F8 here. It goes to New Zealand as a row, and it comes back as a plot that's grown in a field over there. And those plots look like the plots that we saw first in the first year after New Zealand, after the breeding process, we only have one small plot, and we grow a bunch of Czech varieties in the field. So that's ultimately the entire breeding process up until there. Over when I talked earlier about disease nurseries, which Isabella actually runs here with our program, over in this side there's about 5,000 to 10,000 rows because we have genetic studies with students. So graduate students will have populations growing in a rust nursery, and the data that the students collect will form part of their thesis as we try and map genes associated with rust resistance in this particular field and have populations growing there. We also have all our material that's in the first generation of yield trialing tested for rust here, and we score that later in the field and relate the rust scores to what it looks like in the field. If a variety at this stage, the F7 first yield trial, if it has susceptibility to a disease, we cull it immediately. If it falls over in the yield trial, we cull it, and etc. We're growing the exact same nursery that's there of the 5,000 lines. We're growing that in Creston, BC. We're growing that in Manitoba for FHB, Creston for yellow rust. We have another nursery over in another part of the field for bunt and for leaf spotting diseases. And all those disease data inform, before we even harvest our field, the first field of yield trial, we don't harvest lines that have basically 40% or more disease incidence on their leaf structures. With FHB, obviously their head, etc. So that's a summation of the breeding process prior to the yield trialing that you'll see in a minute.